So this is an introduction to atlases, and as Mike said, we don't have full functionality, but I thought it's still important to introduce some of these concepts. So basically, the atlas is kind of a way to visualize information that's in the sequence in terms of structures. And so we'll talk some about this. Okay, so this is kind of, we're going to duck over the weekend. So we're here now. Actually, this is actually the atlas part. So if you remember, we defined bioinformatics as looking at information flow in biological systems. DNA makes RNA makes protein. So here, the DNA sequence makes RNA, the transcripts, which makes a protein. Now, in biology, here you have, this is a phage. So this is a small viral particle. A bacteriophage, actually, it's a virus that invades other bacteria. So it's not going to hurt humans. But it obviously it's bad for bacteria. And bacteria is kind of a brutal world because for every one bacteria, typically in an environment, you have 10 viruses. And so they're, viruses are always attacking bacteria. And it's, it's a really brutal world out there. So you go from one viral particle to lots and lots of viral particles. And so the first genome to be sequenced, we talked about Haemophilus influenza was the first bacterial genome. The first viral genome to be sequenced was sequenced 10 years before that, actually a little bit before, maybe 12 years before that. And here it is. This is the entire sequence for this virus. Fred Sanger got the Nobel Prize for this. This virus is 5,000 nucleotides long, and it's short enough that you could actually put the whole thing on one piece of paper. Here it is. Here's the entire sequence for the organism. This is a virus. It's pretty simple. And what happens is we did a space every 10 bases, just so that you can kind of see. And this is it. Well, okay, you can see it, but what does this mean? Now, this is one way of looking at it. So now you're looking at the sequence in terms of letters, G's, A's, T's, and C's. So this is a DNA viral genome, and this virus infects other bacteria. It's called a bacteriophage. So another way of looking at it, so this is the name of this organism is VX174. It's 5,386 base pairs long, and that's what this thing that you saw here, 5,386 characters, okay? It's 5,386 base pairs long. Now, see there's these sites, and these are called restriction enzymes. Now, this is a map that they made of this in the 1980s. What happens is you can cut with these enzymes, and then you have smaller pieces and you can sequence those and stitch it back together. So that's where this came from. So it's just looking at the genome again. And now this is a different kind of information. Where do these enzymes cut? But you could also have, of course, remember this is a DNA genome, and the DNA gets transcribed into RNA, which the red is the RNA, and the RNA then gets translated into proteins. And here's your proteins, and they're cleverly called P1 for the first protein. So we're here, protein 1, protein 2, P3, P4, P5, P6, 7, 8, 9, 10, 11. That's it. So, and then it's 9, and then they discovered another 9, so it's 9D. and there's a lot of historical reasons for this, but so you could see that basically you have the DNA sequence that gets transcribed to RNA. And you see there's different RNAs here that basically you can start here, and you have one that goes all the way around completely. So the outer one is an entire copy. You can also make part copy, and it stops there. Or you can start here and make a copy there. And so basically you have these different transcripts from the same DNA sequence. Now here's your origin of replication here, which is something different as well, but and so again, this is a circle because it's a circular genome. So this is very different than this. Here's your sequence. That's your sequence. This is the same thing, but now it's just showing it as a circle. You could put all the letters here. You would never see it, but you can start here and you go all the way around. And these are restriction enzymes. And this is looking at transcripts and proteins. So it's all kind of various ways of looking at the same thing. And so when we're developing these atlases, we could make an atlas of this VX174. And so we could look at how much G, A, T, and C you have here. And what you see, and so I just went from 0 to 40%. And so if you just look at these outer four circles here, so the outermost circle is G, and you can see not a whole lot of G's there. And you can see some A's here, but if you look at T's, you see an awful lot of T's. You look over here, this is mainly T's. And then not as much C's. And so this is looking at the skew a little bit of A's and T's. So it's, see here you have more T's and A's, or more G's and C's. And so that's looking at the skews here, but then this is innermost is looking at AT content. And you see this is much more AT rich. Well, so, okay. Now, again, I'm going to back up here. So here, now that you know that, you can look and say, well, yeah, there is quite a few T's there. But again, this visualizing is just kind of a way of looking at all that information. So what we're doing is we're looking at the sequence as we go on, all 5,300 bases of it. And then here's all the genes as well that kind of marked there. And so this is kind of visualizing the whole genome. Now, of course, this goes from 0 to 40%, but what if instead we said, how does it differ from the average? And so if you compare this to this, first of all, this is not as bright, but now we're looking at things that occur more often than average or less often. So its average is gray. And one of the ways of visualizing things is you don't want to see. So your brain, you're not overwhelmed with all this stuff that's average. And so now if you look at the T lane, there's like a few things that are a little surprising, but not that much. And so now this is kind of a way of letting you see, and you look over here and you say, well, something's going on here. There's a lot more G's, so there's a bit of a GC skew. There's a little bit more AT rich here. And so by looking at this, you can begin to see where are things that differ in the genome. And sometimes that's important. And so a big part of visualization sometimes is to take the average and make it kind of disappear because you don't really care about the average that much. So again, here's the whole sequence. And this is just kind of a way of looking. Okay, so we talked earlier about oligomer skews and about structure some and about bases and things. And so I'm going to talk about some of these structural parameters that we use. There is some atlases you can make what's called a structural atlas, or you can make an atlas basically with some of these properties. And so one of the properties we can look at is we can look at how easy it melts. We can look at how curved is it, and we look at something called anisotropic flexibility. So I'm going to talk about those three structural properties, and that's going to be basically the gist of the lecture. Now, one of my favorite DNA structures, is curved DNA, and this is kind of fun because this structure was never really predicted. It was discovered by accident. So probably many of you have run gel electrophoresis. And what happens is you put a sample in a gel, and you turn it on, and there's a current that goes across the electrode. And DNA, remember, is kind of negative charge, so it's going to run towards the positive here. So it's going to run into the gel. Now the gel is kind of this matrix that's got, it's got a lot of instructions in it, and the DNA has to kind of snake around into the gel. And basically the shorter fragments run faster, and the longer fragments run slower. And this is from a gel. This is from probably, 30 or 40 years ago. This is part of my thesis project. But here's a 490 base pair piece of DNA. And it's running in an agarose gel, as it should, as about 490 bases. But in an 8% acrylamide gel, it runs as if it's more than 1,000 bases. So it's running anomalously slow. And it turns out that as you increase the percent acrylamide, it runs slower and slower. And that's because this piece of DNA is curved. And you can have something like this, it's kind of a stable curve, or you can have straight DNA, right? And so if you look at agarose, basically this is in an agarose gel, you have it's this matrix, and the DNA can go through there. This is about 10 nanometers or 100 angstroms. And 10 nanometers, the DNA width is about 2 nanometers. And so DNA can go through an agarose gel fine. And if it's curved, this is okay. The acrylamide gel, particularly at 8%, now the pore size is getting so small that it's basically approaching the size of the DNA double helix. It's in the range of a few microns instead of 10 nanometers. So DNA is about 2 nanometers wide, or 20 angstroms, 20 atoms across. And the higher acrylamide gels, the pore size is so small that basically the DNA, if it has a curve to it, starts running anomalously slow. So where does this curvature come from? And again, no, I got caught without my chocolate bars. That's a horrible feeling. So if you think about Swiss chocolate bars, and the base is stacking on top of each other, they can tilt or they can roll. And so if you have a helix, if they tilt, that's going to change the direction of the helix, and the same with the roll. And if these tilts and rolls, if they're random, you get something like this. But if they're phased with the turn of the helix, you can get a smooth curve like this. But sometimes they can actually be anti-phase, so every 16 bases, and now you get something like this. This runs anomalously fast, this runs anomalously slow, and this runs normal. And so it turns out that when we started looking at some of these DNA that was running anomalously slow, we found these A tracks. This is a bit of a simplification, but basically, if you have an A track every 10 bases, that combination tilt and roll, that changes the helix. And if you do it right, you can get a stable curve. And so curvature is just one out of many type things. You've got curvature, and you have lots of other structures as well. But so the curvature, and on the atlases, we color this so that it's more blue if it's more curved. Just and don't really worry that much about this. So basically, if it's bluer, then it's more curved. And then if it melts more readily, and you can actually calculate how much energy it takes to open up the helix. Remember, we talked about stacking on top of each other. And so in this case, and again, these numbers depend on the particular sequence and what you're looking at, but this is about 7 kcals per mole compared to here's 9. And so the average is going to be 8, right? So 7, 8, 9. So this takes less energy to open it up. So it's more red because it'll melt more readily. So now if we can take curvature, we plot it as we go again, looking at translation starts. So here's where your genes start, your AUG. And so here's the coding region, here's upstream. You see a huge region upstream that's more curved. And this kind of helps the DNA to wrap around the polymerase. And you can look at some of these different structural parameters and you can see that here's the AT content that we were talking about before. And so it's a little more AT rich upstream than downstream. And the curvature, there's a long region that's curved upstream. And that, like I said, plays an important role. So again, this is the same virus we were looking at before, FX174, 5,386 bases long. And now what we're doing, this is called a Genome Atlas. And what we're looking at is we can look at the curvature here. So you can see these little blue regions that have curved. And you can see regions that will melt more readily. That's the red if it melts more readily. Here, a few regions that will melt. And then this position preference, which I'll talk about that in a bit. And then we can look at some repeats and other things throughout this. So whenever you have a piece of DNA, and in the piece of DNA is a coding region, CDS. So this codes for a protein, okay? And so would be where your protein is. AUG is your first one that codes for methionine. Now, you have something called the +1 where transcription starts. And between +1 and where your translation starts, where your first codon is, this is called the upstream region or untranslated region, 5', because it goes 5' to 3', 5' UTR. This is called untranslated. And this is the 3' UTR, untranslated region. So here you've got what's called a TATA box, and it's TAAT. And again, this TA is really, really conserved, and this is not, it depends on how you do it, but basically, this is the TATA box, TATAAT. That's the -10, you have -35, which is TTGCA. And this is the region. Now, if you look at this is actually, so if you start here, one turn of the helix is 10 bases here, right? And then here, the -35 is actually one turn is 20, 2 turns is 30, 35 is 2.5 turns. And what happens is that the helix will come and the polymerase comes and it binds, grabs the -35, -10, it twists it, and it pops it open right here between this TA, because remember, pyrimidine purine step here. This is very unstable. It melts right there, opens it up, and that forms a little bubble, and here's where you start transcribing here. So and you have at the end sometimes a little stem loop, an inverted repeat that'll form a stem loop structure. So this gets translated. And so here you have your 5' UTR. Here's your coding region, your 3' UTR, and that includes sometimes a stem loop structure, Okay, and so upstream here, now you're going to have regions that will, this melts, between here is going to be rigid, and this region is going to be curved. Okay, and so that's because this region here that curves, these interact with the alpha subunit of the beta-beta prime of the polymerase, the RNA polymerase, and it wraps the DNA around. So the curvature really helps this. The sigma factor binds -10, -35. And so you have transcription here where it starts. And so the sigma factor has a lipid basically that will open up the helix and start transcribing here. And so this is kind of a cartoon from a text. So here's your -10, -35, and it opens up and starts forming here. And the polymerase, the DNA is wrapped around the RNA polymerase, and now you have messenger RNA. And so this region around here needs to be flexible and it needs to be curved. Is what's going to happen is it's going to wrap around your polymerase. So it's going to be more blue here. And the region, this region right here needs to be more rigid. And so that's going to be more purple. And stacking energy is going to be more red. And so this kind of signal, if you're zoomed into the atlas and you're looking at the level upstream of a gene, you can have a curve followed by something that's rigid and will melt, that this is a structural signature for promoter. That's kind of how you know. And so here's an example of an atlas. This is called PVIR. There's another plasmid that's responsible for virulence in Campylobacter. And you can see this little structural signature here. So you see rigid, melt, curve. And actually, generally the curve, yeah, you can see here curve, this will melt and it's rigid. So this is a good kind of structural signature for promoter. That's kind of how you read these. This is looking upstream of the ribosomal RNA, and you can actually do some models where you start supercoiling the DNA and you get different promoters to open up. I'm not going to talk a lot about that, but this is the plan. So you can also build these atlases. This is for Ebola virus, and these are the different proteins here. And now this is an RNA virus, so it's kind of an atlas looking at, and we're looking at particular in palindromes because RNA viruses, you need stem loop structures. And you can see some regions of this virus are more variable. So what we've done is we've taken a whole bunch of genomes and said, what regions are the same, it's gray, or do they vary, it's red. And we can look at combinations, some of the regions that melt more readily vary. And this GP is a glycoprotein. And you look within the glycoprotein, you can see quite a bit of variability there. And this is a surface protein, so that makes sense. But you also have some stem loop structures here and some different types of things. And so you can look at this and begin to predict some same things. This is for, the coronavirus, for COVID-19. And you have ORF1AB, you have then these other smaller proteins at the end. And you can look at AT content and inverted repeats, and you can look again at variation here. Some regions vary more than others, and you can look at that. Okay, so now we're going to talk about chromatin again. And so the DNA or RNA in the cell is compacted around 7,000-fold. So for eukaryotes, it's mapped around histones, and these form stable. This is a dimer, a set of 4 dimers. It's an octamer that basically wraps the DNA around it twice. And so here's your chromosome. And if you look at the chromosome actually has, and the chromosome is 1.4 microns. So you can see this with the microscope. And out of that 1.4 micron, basically there are these little fibers that are around 700 nanometers. So that's the width of this. So that's half this width here. It's 1400, this is 700. So this is called the 700 nanometer diameter. This is a kind of coiled coil. And these are wrapped up into this 30 nanometer fiber, or 300 nanometer collapsed fiber. Here's the 30 nanometer fiber. And the 30 nanometer fiber is a bunch of basically, you see here these histones that's wrapped around twice. This is called beads on a string here. So you've got the histones, this histone octamer, the DNA is wrapped around twice. And this is an 11 nanometer fiber. And then the DNA is 2 nanometers, it's 20 atoms across. And so the DNA, the way that it's compacted is it's wrapped around this histone octamer. And these histone octamers then kind of collapse down. They're wrapped in this 30 nanometer fiber, which gets coiled in a 300 nanometer fiber, which is 700 nanometer, which is there. So this is how the DNA is compacted. And so the histones, you've got two H4s, two H2As, two H3s, and two H2Bs. And these are all, these proteins, they form this octamer, and the DNA wraps around these. And what you can do is you could actually take the chromatin and get it so it's called beads on a string. And so you can chop it here with a nuclease. So now you get these pieces that have roughly 200 bases of DNA wrapped around. And then you can do a little bit heavier nuclease digestion, and you get around 145 bases. So 145 bases for DNA, 150, 145 bases is what's called the persistence length for DNA. And that's persistence length is if you have like a metal rod and how long it would take for it to bend around on itself. And it's kind of a well-known structural parameter. And for DNA, it's around 145 bases. And that number often comes up for DNA. And this is basically, so this is in the case for eukaryotes, wraps around histones. In bacteria, 145 bases is what wraps around polymerase, wraps around other things. So that number is kind of an important number. And it's roughly, if you will, around 15 turns, 14, 15 turns of the helix, double helix. One turn is 10 bases, about. Okay, so what Andrew Travers did, so Andrew Travers is a scientist working, he's a professor at Cambridge University. I think he's retired now, but I worked with him in the late 80s, early 90s. He's very much a structural guy, and so this is with Steve Satchel and Horace Drew, and they published a paper in 1986 looking at sequence periodicity in chicken nucleosome core DNA. And what he's looking at is he trimmed it, like I showed you, those 145 base pair pieces, and then he sequenced them. And again, this is the 80s, so sequencing was kind of expensive. But what they did is they're looking at, they know where it starts and stops, and they can look at whether these trinucleotides are facing outside the nucleosome or inside. And they kind of scaled it, and they counted, basically, the sequencing counted how many times these trinucleotides occur. And so they say these guys, basically 19% of the time they're outside, they're facing out, and then this is the most abundant, and then it goes down to around 0 here, and then it goes more abundant, and these guys 28% of the time they're facing inside. So you're just looking at these trinucleotides and how they occur as it's wrapped around the double helix. And so around 96, this is when I first started working with people in Denmark, and basically what they're interested in, I'll explain this in a minute, but basically this is Pierre Baldi and Søren Brunak and Yves Chiron and Anders Krogh. They basically are looking at these regions with low position preference, because what they're really interested in is nucleosome positioning sequences. So these guys, they have a high position preference. By that they mean either they're always going to be facing outside or always facing inside. And they're often found in nucleosomes, but it turns out that these guys in the middle aren't found in nucleosomes. And the reason they're not found is that there's some properties for them. They don't bend very easily. And so these guys in the middle with a low position preference, that's what's called anisotropic flexibility. And that's really important. And so basically, again, with these structural things here, now we're looking at this is kind of the flexible DNA, if you will. So curvature is more blue if it's curved. And this is something called position preference, because it has to do with nucleosomal position preference. Sometimes I call it Travers for Andrew Travers, but basically it's these numbers. And so if it's green, what it means is it doesn't like nucleosomes. It tends to be excluded from nucleosomes. And I'll come back to why that's important. So if it's green, it has low, basically it's more rigid. It's not going to wrap around nucleosomes. If it's purple, it's what's called anisotropic flexibility. You can bend it a certain way. And then if it's red, it will melt more readily. And so one of the things we could do early on, and this was so let's see, this was 20 years ago, good grief, published this in Nature, and just looking at POMB. So at this time, this is like the third eukaryote to be sequenced. And Paul Nurse was the main author on this paper. It was kind of fun because he got the Nobel Prize as he was writing this paper. And being one of the co-authors on there, it was fun watching him he got a little bit, he was very proud who got the Nobel Prize. I'll phrase it that way. But anyway, some of his comments. But so one of the things we could look at is we can actually map the 3 chromosomes here. So Pombe only has 3 chromosomes. It's almost 12 million bases total. So here's chromosome 1. It's 5 million bases long. Chromosome 2, chromosome 3. And we can look at repeats, and there's not that many repeats. This is in a yeast chromosome. And then we can look at AT content, and the ends, the telomeres tend to be more AT rich, and this is the centromere here. You see there's not any genes, and it's more AT rich. This is centromeric DNA. And then here's this thing called position preference. And when we map this, we see that there's some regions here that are more green. And in fact, see this guy right here, this guy is really pretty green, and we can zoom in on this. And there's this gene. And so one of the things when I was building these atlases, and I was talking with Val, so Val's the main author, the lead author on this project at Cambridge, where it's sequencing it. And I said, what is this gene? Because according to my predictions, it might be highly expressed. And she writes back and says, yeah, Valerie Wood, she says that this is one of the most highly expressed genes in Pombe. She says, why do you ask? How did you know? And I said, well, because according to our predictions, this gene, basically, if you look at the trimers in there, some of them, they don't like to be wrapped around nucleosomes. So they're going to be free. They're not condensed. So they can be highly expressed. And it turns out this is a very good way of actually predicting highly expressed genes, just looking at it. It's very kind of naive, very simplistic, but it works. And it's actually pretty fast. So you can look at repeats as well. So we have direct repeats, we have inverted repeats, and then you have something called mirror repeats and everted repeats. So we're not going to really look at that. So direct repeats, again, are blue, and inverted repeats are red. So now here's the Genome Atlas. And so this is, and in fact, this is Karsten. This is back 21 years ago. So Karsten, this is part of his PhD project. And this is kind of fun because what we could see, this is a plasmid for coli. And we see that here, this region, it's very AT rich. The structural property is a little bit different, and it's flanked by inverted repeats. And these are actually IS elements. And so without really doing anything, just looking at structural properties, we say something's going on there. Well, this region right here, it turns out, is a hemolysin toxin. And it turns out that you can use these atlases for finding, and it's flanked by these IS elements. And you can look for novel toxin genes and other stuff, and they kind of line up for structural properties just from this. So this is it's the same plasmid, but from kind of 2 different isolates. And this is, and they started in different places. If you can imagine taking the bottom one and rotating it around, you can see that it matches. But this is, again, just kind of a way of visualizing what's going on for the genome sequence. And so this is now the whole E. coli genome. And so you have these structural parameters on the outside, and here are your genes. And then now you're looking at repeats here, and then you're looking at GC skew and the AT content. And this is kind of looking at it all at once. And this is a big thing. So it's 4,000 proteins, 4.6 million bases, and it's just kind of a way of visualizing chromosomes and what's going on. And you can compare this. So this is the first Haemophilus influenza genome that was sequenced. And then this is another one, and you can kind of compare these two, and you can see differences between these, looking at it. And so what we could also do is we could look at, this is what I call my Dogma Atlas. This is published back in 2001, and the idea is you have your DNA here, and we did some experiments with this particular E. coli. This is a common lab strain. We grow it up, we grow up 10 mils, we take 5 mils, we look at the RNA concentration, which is a green. And at that time, this is, again, 20 years ago, right? We were using microarrays. And the proteins were using proteomics, and we could look at the protein concentration from the same sample. And generally, there's an agreement. You have RNA and you have protein. But sometimes you have RNA and no protein. Here you have lots of RNA and no protein. So who should I pick on? Kaylee, can you tell me why you'd have RNA but no protein? Is anyone still there? Is everyone asleep? Andrew, are you there?
Yes.
Yeah, we're still here.
So Haley, can you answer the question, do you think, or not?
I don't think so.
You don't think so? That's okay. Andrew, do you want to try?
What was the question again?
Yeah, see? So if you look over here, can you see my mouse?
Yes.
So this green, this is lots of RNA, but there's no protein. What do you think is going on?
It's probably not TR, or it's probably not protein coding.
It turns out this is, but you're right. It could be ribosomal RNA, and then that would make sense, right? But this actually does code for a gene. And this down here is also. And you can see this guy and this guy. So you can see a lot of places. It turns out that, so the kind of in biology, you think, okay, if we know the transcript level, we know the protein level. And it's generally true, like 85% of the time it's true, but it's not 100%. And sometimes, for a variety of reasons, your RNA can be stable, your protein's not. It depends on when you do this. RNA, the half-life for RNA is only about 2 minutes. A lot of these people, they're worried about getting the vaccine, the Pfizer vaccine. Oh, it's RNA. It's like, yeah, it's the reason you have to keep it at -70 is because it's not very stable. RNA, it disappears really, really quickly, generally. Sometimes it lasts around a while. But depending on how you do it, now this was done by a postdoc who works with RNA. He's really very careful. And so he basically kind of locked it in early. But so you can have lots of RNA, but maybe the protein's not stable. Whoops.
So
okay.
So what that's showing is that the RNA just isn't getting the ribosome's not getting to it fast enough, I guess.
It's not being translated, yeah. And so you could see over here you have protein but no RNA. Let's see if I can find another example. There you see protein but no RNA. Here, see that? You see quite a bit of protein but no RNA.
Right.
And that's because in that case the protein's more stable. And you see over here this is OMP. So these outer membrane proteins where they can last, these proteins can last 24 hours or longer, and the RNA can only last a few minutes. So sometimes you have lots of RNA, and you have lots of protein, and that makes more sense kind of that you can have protein but not RNA. So the point is it's not 100% a one-to-one correlation. 85% of the time it's not bad, but it's not. Sometimes you need to take that into account when you're considering things. Okay, so last bit, we're just going to talk about chromatin. So here's a picture of E. coli. And now if you're going to take one of these E. coli cells and break it open, this would be this length of the cell. And this is all of the DNA that's in there. And you look and you can see it comes out and comes out. So remember, the DNA is compacted 7,000-fold in the cell. And so you have lots and lots of DNA. And so it's compacted a variety of ways. You have something, see these kind of supercoils here, it's called plectonymic supercoils. This is like a rubber band. And you have something where it's wrapped around a protein or something, or nucleosome or something, that's called toroidal supercoils, because it's wrapped around the toroid. Now, one of those toroidal supercoils, of course, you could have RNA polymerase that wraps around the RNA polymerase. And at the edge here of this, you have curved DNA, and they tend to be located at the end of plectonymic supercoils. This is curved DNA there. Now you also have a topological boundary. So this would be attached to the membrane or something. So the DNA is actually physically attached. And so basically the DNA is supercoiled. And remember, it's highly compact, and you have this kind of dynamic thing. And so these toroid supercoils, you have these things that bind DNA and will compress it. Now in eukaryotes, this is well known. In chromatin, you have things, if they're wrapped up in chromatin, they don't get expressed. And if they're chromatin free, they are. That's this whole idea of what we're looking at with that green on this nucleosome positioning, right? The question is, does this work in bacteria? And it turns out it does. So this green here, this position preference, you can see here, you can see where the green are. These actually correspond to highly expressed regions of the genes here. So these are highly transcribed. So this is RNA levels. And you can see sometimes the RNA levels and the protein levels are pretty high. And in particular, if you look now at these chromatin proteins, so these are integration host factor or FISPI, these are chromatin proteins in bacteria. And where they bind here, that's the red here, here, here. You can see where they bind, you don't see highly expressed regions. And so here you see highly expressed, and then you don't see it expressed. There's chromatin proteins. The absent of chromatin proteins, you highly expressed. Chromatin proteins, it's not there where the binding sites are. Expressed, it's not. So you can actually see that the chromatin is playing a role in gene expression. That's just kind of an interesting thing. And so the point is that bacteria have chromatin as well. Is the thing. Okay, so I have just a couple of questions and we'll be done. So this is a goose. So you have these two sequences, and here's your question. Which sequence is going to melt first?
The top one.
Try again. Well, they have two choices. But the top one's 100% AT rich, right?
Yeah.
Wow. So what's going on? But look at this. You have remember we talked about purines stacked on top of each other? So every AA, here you have 5 kcals, 5 or 6 kcals every time. So that's 5 plus 5 plus 5. So you're having a lot of energy here from stacking. The most important thing is stacking, not hydrogen bonding between them. So you get 3 kcals or 2 kcals for an AT pair, 3 for a GC, right? But here you're getting 5 or 6 for an AA stacked on top, and on the other side you're going to have AA stacked on top. Here, if you have a pyrimidine purine step, and these tend to be not very stable at all, and you get penalized for that, and you only get 3 or 4 kcals per mole stability from that. And so from a thermodynamics point of view, this is far less stable than this. So it's not just AT content. Now, this is a bit of a straw man argument, a cartoon example, but it's something we're thinking about that because normally you don't have long stretches like this. This is more synthetic. But the point is that it's not just whether it's AT rich or not. You need to look for pyrimidine purine steps. That's why the TATA box, TATAAT, that will melt more readily. Not only is it AT rich, but it also has these pyrimidine purine steps. And that's what's really the most important thing from a thermodynamic point of view. Okay.
Sorry, can I ask a question?
Yes, of course.
So normally, like when you design primers and you're looking at the DNA, it's normally you try not to get too many G's and C's because it makes the melting point higher.
Well, kind of, and this has gotten into this is again urban legend. A lot of people believe that, and it's kind of partly true most of the time. Just like 85% of the time, you can predict the level of proteins from the transcript.
Right.
Right. But it's not always true.
Okay.
Now you don't need to do rigorous. It turns out that you could actually do some fairly quick calculations without. So some people they do all these melting calculations and it takes hours and hours to do for a little 20 base pair primer. That's stupid. There's some things you can do that take just a few seconds that can look at that you can predict. So if you're interested, we could talk sometime about how to better design primers. You want to avoid G's in a row if you can.
Right.
In primers.
Yeah. I normally just use like NCBI, and they do it all for you. Is that?
They do. It works most of the time. It's not the best, but it works okay. It works okay. I mean, it generally it'll work. But there's some other things to take into consideration in primers, and it's interesting because there's a lot of assumptions that kind of get in it, and just like in the human genome, they think they have this 100 base pair piece, they're looking for SNPs. Right, and they'll do a probe or something, and they say, "Oh, how often does this 100 base pair piece occur in the human genome?" What they don't realize is that the human genome is mostly repetitive DNA, and so sometimes this 100 base pair piece is going to be found 3 million times in the human genome.
Right.
And so what they think is SNPs is they're just looking at variants. It's not really. So there's a lot of things to consider in primers. It's a bit messy. A bit of an art. And sometimes with the primers, you need magnesium. It depends on the magnesium concentration, and all the other stuff. It's like, it's almost like it's more artistic. It's like cooking in a sense, right?
Yes.
some people are better than others, and it takes a bit of skill. So that's kind of a cartoon example, but that's making a point that you have something that's 100% AT rich, and it melts less well than something that's only 50% AT rich. So, okay, I talked some about this. You've got, I don't know what's going on. My siblings are all of a sudden, I have 3 brothers and a sister, and also they started texting me about their dogs or something. I don't know. But anyway, sorry, got distracted. So you have, I talked about using DNA structures to predict promoters. Do you guys remember this? There's kind of 3 parameters that you look for. Anybody want to guess? Do you remember you see if it's curved? You have this kind of structural signature this is right at the promoter region now. So if you're looking at a big genome, you're not going to see it. You have to zoom in at the level of looking upstream, right at the -10, -35 region, and that region upstream. So you have a curve upstream. So -50, -100, you've got this curve region. So you have curve, then you have a rigid region, and something that melts. Curve, rigid, melt is kind of the structural signature for promoter. Okay, so now I talked a little bit about this gene expression, and you have a gene, and you want it to be highly expressed. What could you do? There's a couple of things from the lecture that you could do to make it more likely to be highly expressed. Any suggestions?
A strong promoter.
A strong promoter, and how would you do a strong promoter? What does that mean?
Like something that is, like highly regulated at that promoter region.
So what about the previous question? Do you remember the previous question there?
About the structures.
Yeah, so obviously that's one way. So you could say you look for something that's curved, rigid, melt. So you could look for that kind of structural, you could actually design it so it's that way. Another thing is if you put it close to the origin of replication, it's likely to be highly expressed, because genes generally closer to the origin are highly expressed, and in the terminus they're less highly expressed. The final thing is, remember this chromatin? Remember there's this thing called position preference? And certain trinucleotides don't like to be wrapped around nucleosomes. So if you could engineer that into the gene so that it's not compressed, then it can be more likely to be highly expressed.